Hi, everybody. I'm really grateful for this opportunity to present. I'm grateful to the organizers for letting me do this virtually, and I'm also grateful to all of you for your willingness to listen to me. So here's the title slide. I want to tell you about golden moles, mostly in the Chrysochloridae. It's a small insectivorous grade clade of Afrotherian mammals. And it's based on this paper that came out in print a few months ago, Bronner et al., in the Zoological Journal of the Linnean Society. To start my talk about golden moles, I want to first tell you about their sister clade, the Tenrecs. And I think in this crowd, Tenrecs don't need too much of an introduction, but suffice it to say that they're very diverse. There are over 30 species, most of which are on the island of Madagascar. There are Tenrecs that burrow. There are Tenrecs that from a distance look like shrews or hedgehogs. There are Tenrecs that kind of look like a mix between an opossum and a raccoon. There are semi-aquatic Tenrecs, but they're not raccoons or opossums or shrews. They're their own unique radiation, mostly on Madagascar. There are a few continental African species, just three actually, and all of three of which are semi-aquatic, but they're not related to the semi-aquatic Malagasy Tenrec that's among the microgales. The golden moles, compared to the Tenrecs, are less diverse ecologically. There are about 21 species, and most of the species, all extant, are in and around the country of South Africa. There, you could see, I hope you can see these little polygons. That represents the occurrence of the various extant species, and you perhaps note that there are some of these polygons in the tropics. But the number of species, it's really just two of them way up there in the tropics, and most of them are down here in South Africa, and all of them are ecologically limited to burrowing, right? There aren't any semi-aquatic or gliding or other kinds of adaptations that you can see among the Tenrecs. They're all pretty much non-visual burrowers, as you can see in these images, and perhaps the most famous golden mole is this guy, Erametopa grantii, most famous because there's that lovely clip of Grant's golden mole in The Life of Mammals, now almost 20 years ago. So quite a few years back, published in 2010, some colleagues and I got together what today is a very small data set to try and work out the systematics of golden moles. And it was a very small data set, like I said, 800 aligned bases, part of one exon of one nuclear gene, growth hormone receptor, plus a morphology data set. And interestingly, we were able to get a fully resolved topology for the Tenrecs, and you can see quite a few of these 1.0 posterior probability support indices, which have actually withstood the test of time. I mean, we weren't the first ones to suggest this branching topology, but much bigger data sets have been published since then, which support a similar topology. But we weren't able to work out how the earliest branching events within the golden moles looked, right? We weren't able to get a bifurcating tree for that first branching event here in this red rectangle, and not in the second one either, as you can see. So that's a problem, and we wanted to work on that. So my colleagues and I, led by Gary Bronner, University of Cape Town, got what still is, by today's standards, a very small data set, but it's a lot bigger than the one we had back in 2010. So 3 mitochondrial, 3 nuclear genes, about 6,000 base pair alignment. We did have a morphology data set. I'll get to that in a moment. Here's just one of the tables showing you our sampling. So it's not a huge data set, but a lot bigger than what we had. And to cut to the chase, we still couldn't figure out the root of golden moles. Now, the topology I'm showing you here on the right is one of the figures from Bronner et al. 2024. And it shows you a fully dichotomous tree, but that support index on this first divergence within crown Chrysochloridae that I'm circling is pretty low, and that's really not something to hang your hat on. Interestingly, I think it's interesting anyways, our data set for the Tenrecs, or as applied to the Tenrecs, the larger data set pretty much supported all of the nodes that had these posterior probabilities of 1.0. So you don't necessarily need a huge data set to get a dependable result, at least one that stands up to further scrutiny. But we still have work to do with this golden mole group. We don't have a well-resolved root node, or at least earliest divergence within the crown clade. Okay, so this brings me then to the somewhat odd title of my talk, One Bone Can Do It. I'm being a bit cheeky here and speculative, but I think the anatomy of this bone, the hyoid dentary articulation in golden moles, sheds some light on what the earliest divergence point within golden moles really is. And the story here begins in another paper by Gary Bronner, way in 1990. He noticed for the first time, I think, that golden moles have this unique feature. They have a lower jaw that actually articulates with an articular facet and everything with the hyoid apparatus. This particular element is called the stylohyoid, and this illustration shows you in an amblysomus, one of these South African golden moles, this dentary hyoid articulation. And Gary went on to describe the hyoid apparatus. You can see the stylohyoid elements here, which if you know anything about hyoid elements, these are bizarre. No other vertebrate, at least no other mammal, has an element like this. That's the articular facet for the jaw. And this jaw articulation between the stylohyoid, or this dentary stylohyoid articulation, it's the rule within golden moles. At least all of the species that Gary looked at back in the early 90s had it, and that's what I'm circling right now in a stylohyoid element of an amblysomus gunningi. So he didn't have, for a variety of reasons, data on the hyoid apparatus in a couple of these golden moles, in particular Eremetalpa, Grant's golden mole, the famous one from Life of Mammals, and one of the tropical golden moles, what we now call Huetia leucorhinus, used to be in the genus Chalcochloris. It's pretty certainly in Huetia. You can see that Huetia has a much more primitive stylohyoid element that I'm pointing out with the mouse pointer, and its angular process of the dentary is much more primitive. What's more, compared to these other golden moles like Chlorotelpa, Amblysomus, and indeed Eremetalpa to some extent, is the anatomy of this angular process of the dentary. So another really important development for those of you interested in golden moles was the publication in 2015 by Martin Pickford, describing what are to date the best fossil, or the anatomically most complete fossil golden moles we yet known. And that here is a picture of two specimens, a skull somewhat banged up, but still anatomically well represented, Namachlorus arenatans. And I hope you can just make out the angular process of Namachlorans. If it's not evident in that slide, it should be in this one. It is relatively primitive. It looks like what we have in Huetia, not what we have in, say, Amblysomus or Chlorotalpa or most of these other species of golden moles. Now, the age of the fossils from this site in Namibia, what Pickford argued, he said it was Lutetian. I think they're a bit younger, could be as young as even into the upper Oligocene. I mean, you can look at, we cite papers by, Marivaux and, Tabus et al. And Seifert and Salam. That's a whole nother story that I'm not going to get into, the age of this locality or these localities in Namibia. But there's no question that the fossils are really nice, and they show us a lot about the anatomy of fossil golden moles somewhere between, I think, 30 and 20 million years ago. Okay, where was I then? The point here, though, is that Huetia, this tropical golden mole, and Eremetalpa have the most gracile, primitive hyoid apparatus of any chrysochlorid. And indeed, Huetia has an angular process that is really close anatomically to that of this Namibian fossil Namachlorus. Now, there's another interesting sort of anecdote, speculative anecdote on my part that I want to throw out there, which is the fact that one of these species that's reconstructed basal in our DNA insertion deletion topology that I'm showing you here, Huetia leucorhinus, is only known from the tropics. It's not from the southern tip of the African continent. In fact, it's just these two species, Huetia leucorhinus and Kilometalpa stuhlmanni, who occur above the 18th parallel, right? All these other species are below that. This kind of raises the possibility, and again, this is speculation on my part, that the areas where we find the extant diversity is not reflective of where the clade originated. Long story, I think this is worth looking into. I think it would be worth considering better sampling from these tropical regions. I mean, golden moles are not obvious animals. You can literally stand 50 centimeters above one in a burrow and you never know it's there. And that just becomes more difficult in the parts of the African continent that are tropical and they're less accessible to, sporadic human searches. Okay, so just want to throw that out there for consideration. Another sort of speculative point that I want to make, which is, well, it's a prediction really. Another thing about golden moles is that they're rather conservative in terms of their karyotype. Almost all of these species, well, at least most of these species of crown golden moles that I've showing you in this red box here have a karyotype, that is the number of chromosomes in their diploid cells. Usually it's 30. Whereas if you compare with the Tenrecs, they're all over the place. Karyotype in geogale is 14. It can exceed 42 in some species of microgale. And yet the golden moles are much more conservative, except for Eremetalpa. This has the lowest karyotype, 26 in Eremetalpa of any golden mole. The karyotype in Huetia is unknown, but the prediction based on this topology is that it's going to resemble that of Eremetalpa more so than these other golden moles. And the other thing I want to point out with this figure is just this quite striking difference in branch lengths between the Tenrecs on the one hand and the golden moles on the other. You can look in our paper. We did a time tree. The golden moles are a younger radiation. They took place, or that radiation took place over a much shorter period of time compared to the Tenrecs. And I think that's reflected in the anatomy of the karyotypes as well. Okay, so that brings me to the end of my talk. Conclusions: Eremetalpa and Huetia are probably the most basal species of chrysochlorids. Obviously, that's something to be vetted with larger samples of genetic data from these animals, among other sources of data. The hyoid jaw articulation and enlarged stylohyoid evolved within the chrysochloride crown group, and I think, and I hope I've convinced at least my co-authors too, that is indicative of a basal position for the two taxa. They're not synonymous with their hyoid morphology, but they do differ in having a somewhat more simple hyoid morphology compared to the other golden moles. And we think that's consistent with their position as a sister taxa to other golden moles. And with that, I'll thank you for your attention.